Okay, so now, right, we are going to start with the next video. In this next video, right, so we are going to learn about urine formation. Okay, so in this subtopic, so we are going to see, you have to explain, right, the process of ultrafiltration, right, reabsorption, secretion in the formation of urine. So ultrafiltration maksud dalam bahasa Melayu dia ultra turasan. Reabsorption, penyerapan semula, secretion, rembesan, right? Okay. Okay, so you can see again, so let's recap on the structure of an efferent, right? So you have here the glomerulus and also the Bowman's capsule, right? So nearby to the glomerulus and the Bowman's capsule, you have the proximal tubule, okay? And then following this convoluted proximal tubule, you will have the loop of Henle, right? So loop of Henle divided into two, descending and also ascending. All right. And then you have here, following the tubules, then you have distal tubule, right? And then you have the collecting duct. Okay. And surrounding these renal tubules, you have blood vessels. Okay. The glomerular capillaries. We have the vasa recta, right? And also peritubular capillary. So remember, peritubular capillary is the one that surrounding the distal and also proximal, and vasa recta, blood vessels, those blood capillary that surrounding loop of Henle. Okay, right. Next, we are going to see the process that involved in the urine formation, which involving the first one, ultrafiltration. So the main structure that involved in the ultrafiltration is the glomerulus. All right. And next, following the process of reabsorption and secretion, which happen or involving the proximal tubule, the loop of Henle, even the distal tubule, as well as the collecting duct. Okay, right. Okay, next we go to the overall process of urine formation. Start with the filtration, which we call as ultrafiltration. Okay, so the filtration is the excretory tubule collect filtrate from the blood. So blood coming through the kidney via efferent, arteriole, and it will be filtered here in the glomerular capillary. Okay, within the Bowman's capsule. So water and solutes are forced by few factors, which the main factors over here is the blood pressure, right? So water and solute is forced to cross the selectively permeable membrane due to the difference in pressure, right? And then from the glomerular capillary, it will be filtered into the tubule, into the Bowman's capsule, and then into the excretory tubules. So that is generally what happened in filtration. Okay, next, reabsorption. So reabsorption ialah serapan semula where the transport epithelium of the excretory tubule reclaims valuable substances from the filtrate and return them into the body fluid. So you can see from here what happened. Filtrate ataupun kita panggil bahan-bahan tertapis will be reabsorbed or is reabsorbed from the excretory tubule into the surrounding capillary or into the body fluids. Whereby secretion is the process by which you can see other substances, right, like toxin, drugs, excess ion are secreted from the body fluid or from the surrounding blood capillary into the excretory tubule for excreted, right, to be excreted, or maybe some component need to be absorbed, right, or to be excreted in the urine. And thus excretion is the finish ataupun the end process where the altered filtrate leaving the system, right, through ataupun in the form of urine. Okay, right. So that is the idea on the overall process of urine formation. Okay, Now let's start. The urine formation is accomplished as we discussed earlier. It involved these three mechanisms: filtration of plasma or ultrafiltration. Second one is reabsorption, and the third one is secretion. Okay, right. So now let's go and look at ultrafiltration. So ultrafiltration is the process by which small molecule in the blood, right? So what are the small molecule? So we will discuss later. Small molecule in the blood that will be forced out of the glomerulus, remember, due to the pressure, right? So it is forced out from the glomerular capillary into the lumen of the Bowman capsule to form filtrate, right? So this process is a passive process where it does not require energy. It happens due to the difference in the hydrostatic pressure. Okay, different in pressure. So hydrostatic pressure drive the filtration process to be succeed. And factors that also contribute to the ultrafiltration are, first, the main one, high hydrostatic blood pressure. Number two, large surface area for filtration, luas permukaan tapisan, right? And next one is permeability of the filtration membrane. Ketelapan membrane turasan. Okay, all right. So let's see one by one. Okay, ultrafiltration. So ultrafiltration is a process again where small molecule in the blood like salt and HCl, sugar like glucose, amino acid, and nitrogenous waste pours out from the glomerular into the lumen of Bowman's capsule. Blood cells, right? The white blood cell, the red blood cell, right? Proteins, albumin, for example, right? And large molecule cannot cross the epithelial of the tubule or of the nephron and will remain in the body fluid. Okay, that's why if you are doing what you call it, medical checkup, and the doctors found out that there are white blood cell or red blood cell in your urine, so you have to be further investigated, because red blood cell, or what we call it, white blood cell shouldn't be in the urine. Why? It should be remain in the body fluid, right? So why does that happen, right? So we will further discuss later. Okay, right. Next. So let's see the factors that contribute to the succeeding of ultrafiltration process. Okay, the first one is high hydrostatic blood pressure. Okay, so the high hydrostatic blood pressure is contributed by the features or the characteristics of the glomerulus. Okay, so how does glomerulus maintain the high hydrostatic blood pressure? That is because the different diameter between afferent and efferent arteriole. So for your information, afferent arteriole has larger diameter compared to the efferent arteriole. So the larger diameter of afferent arteriole provide enough high pressure to force the blood out of the capillary of glomerulus. Okay, right. So the afferent arteriole has larger diameter compared to the efferent arteriole. So the blood pressure in the glomerulus is high enough to force the substances, which is the small substances or small molecule in the blood, to be forced out from the blood into the lumens of Bowman's capsule. Okay, so please remember, Bowman's must be spelled as apostrophe S, Bowman's capsule. Okay, right. So you can see from here, this is the picture of glomerulus, where you can see this is afferent arteriole, okay? And this is efferent arteriole. So the blood coming from the afferent arteriole is in the high pressure, right? Which is enough to force the blood out of the glomerulus into the lumen of the Bowman's capsule, and it will be known as filtrate. So what happened to the dose to those unfiltrable component, you can see the unfiltrable component will be sending out of from the efferent arteriole. Okay, right. Second factor is large surface area for filtration. So it is provided by the coil of glomerular capillary. So the glomerulus is coiled and compressed, like what you call it, which providing a very large surface area for the process of ultrafiltration. So thus it produces large amount of glomerular filtrate. Okay, right. So you can see this is the glomerular capillary. All right, so this is a detailed view of the Bowman's capsule. So you can see this is the glomerulus, crumpled brain-like structure, right? So you have here the afferent arteriole, right? And then this is the efferent arteriole. Okay, right. Next, factor number 3 that contribute to the process of ultrafiltration, involved in the process of ultrafiltration, is the permeability of the filtration membrane. Ketelapan membran turasan. Okay, the glomerular filtrate will pass 3 barriers before entering the Bowman's capsule, right? What we see in the animation just now, we can see that as if like the process is very simple. The substances or the small molecule just pass through the glomerular capillary into the Bowman's capsule, right? But actually, if you zoom in into the structure of the glomerulus, they are actually involving 3 other what you call it, barriers before the filtrate, before the component of blood can be known as filtrate. Okay, right. Number 1 is glomerular endothelium. So where is glomerular endothelium? This one. Okay, so this cell, okay, this glomerular endothelium has pores or fenestrations that increase the movement of fluids through it, right, in bulk. So you can see this is the fenestrace, or we call as pore. So with this pore, it allowing fluids to pass through easily in bulk, right? From here, okay, into the Bowman's capsule. So this is the lumen of Bowman's capsule, and this is from the lumen of glomerular capillary. Right? So this is the first barrier, the endothelium, glomerular endothelium. Okay. Second one is glomerular basement membrane. This one. Okay. The glomerular basement membrane is selectively permeable membrane, which allowing, right, which only allow small solutes and water, right, to pass into the Bowman's capsule, but not blood cell. Okay, so this is number 2, right? And then number 3 is the endoepithelial cell of the Bowman's capsule, or that we call as podocyte. So this is the epithelial podocyte. Right? So the third barrier is the epithelial layer of the Bowman's capsule that we call as podocyte. So these cells are called as podocyte because they have food processes, all right, that interdigitate and form slits pore. So you can see they are interdigitate to one another and forming slits, sort of like small space, all right, that make sure that ensure only small molecule can pass through it, right? So these are the third factors, permeability of filtration membrane that contributed to the features or the characteristic of the glomerular endothelium, glomerular basement membrane, as well as the podocyte. Okay, right. So what are the components of the filtrate and the component that exit out of the efferent arteriole, right? The filtrate, maksudnya bahan-bahan yang berjaya ditapis adalah small molecule and water like salt, glucose, amino acid, vitamins, nitrogenous waste like urea, and other small molecules. So don't forget water. Okay, right? And no blood cells and plasma protein, right? White blood cell, okay, red blood cell, right? So no blood cells can be filtered, or it shouldn't be filtered by the glomerulus. Okay, in the efferent arteriole, the non-filterable component are including the blood cell, protein, as well as platelets, red blood cell, white blood cell, platelets, and also proteins. Okay, so these are the content of the glomerular filtrate with the content that exit out of the glomerulus via efferent arteriole. Okay, right. So what happened to the non-filterable blood component? So as we discussed earlier, the non-filterable blood component will exit or will be, entering or transported into the efferent arteriole. Okay, from efferent arteriole. So this is efferent arteriole, and then it will be going into the peritubular capillaries, and then travel into the vasa recta, right? Before exiting the small veins and large renal vein. Okay, right. Okay, so that is all about the first step, or that we call as ultrafiltration. Okay, next we go to the reabsorption process. Okay, what happened during reabsorption? Okay, reabsorption happened, all right? First, we have to know what is reabsorption. Reabsorption is a transport of molecule or any useful substances out of the filtrate, meaning out of the excretory tubule, right, into the surrounding blood vessel or to the surrounding body fluid. Okay, so it is a highly selective process which enable, right, material to return to the blood and leaving waste and excess of other substances in the filtrate to be excreted in the urine. Meaning, let's say useful substances like your body cell need more glucose, so glucose will be reabsorbed from the filtrate into the surrounding blood vessel, right? But if let's say you have excess of glucose, for example, so the excess glucose later will be retained in the filtrate and exit in the urine. So that's why people with diabetic, if you test them with you are doing urine test, so basically glucose can be found in their urine if the person is having a very high diabetic problem. Okay, right. Okay, now let's see, where does reabsorption happen? Reabsorption occurs at the proximal tubule, loop of Henle, distal tubule, as well as collecting duct. Okay, right. So let's see one by one. What happened at proximal, what happened at loop of Henle, distal, and also collecting duct. Okay, right. Okay, so as summary, you can see reabsorption is referring to this process where useful substances is reabsorbed from the tubule, excretory tubule, right, to the surrounding blood vessel. Okay, right. So you can see in the selective reabsorption, some component like sodium, amino acid, glucose will be reabsorbed, right, from the from the filtrate or from the excretory tubule into the surrounding capillary. Okay, right. Now let's see and focus on what happened at proximal and distal tubule. Okay, so you have here is proximal tubule, right? So in proximal tubule, there are two modes of reabsorption that happen, active and passive. So the orange arrow, right, representing active transportation or active reabsorption of substance, right, or solutes. and the purple one is representing the passive transport. Okay, so you can see here, sodium ion, right? One of the sodium ion nutrients like glucose, right, amino acid, usually will be reabsorbed actively. You can see here, Na plus and also nutrient will be reabsorbed actively. Okay, but why you can see here, madam, this is NaCl, but just now you say Na plus. Okay, kenapa? Right, dengar ni. The transfer of positive charge, right, as sodium ion is reabsorbed, it will drive the passive transportation of chloride ion, right? Sodium ion is actively reabsorbed. Chloride will be followed by passive transport. Kenapa? Because it wants to balance the electrical charge, right? So yang ni positif, dia akan tarik yang negatif. Lebih kurang macam tu. Okay, right. So when you have reabsorption of salt and nutrient, remember the surrounding tissue over here will be increased in terms of its osmolarity. Sorry. Okay, this area will increase its osmolarity. Kepekatan kawasan-kawasan sekeliling ini akan semakin pekat. Alright, so to balance that osmolarity, water follows by osmosis. That's why you can see here, water followed by osmosis. Okay, and as for potassium ion, water, bicarbonate ion, it is reabsorbed passively. So you can see bicarbonate ion, potassium ion, and water is reabsorbed passively. All right. Okay, so that is what happened at proximal tubule. Okay. So what are adaptive structure that we can see at the proximal tubule? So you can see here at proximal tubule, it has microvilli. Okay, so proximal tubule is lined with microvilli that will increase the surface area for reabsorption, and it has abundance of mitochondria that will produce ATP to provide energy for the active transportation of solids. All right, and molecule like sodium ion, okay, nutrients, and so on. Right, so you can see it has microvilli, right, and also a lot of mitochondria. Okay, right. So that is the adaptation of proximal tubule that enhance the process of reabsorption at proximal tubule. Okay, right. Next, let's go and look at what happened at the distal tubule. Okay, so reabsorption at distal tubule. So in distal tubule, active transportation or active reabsorption of bicarbonate ion and also salt. So you can see now here at distal tubule, bicarbonate ion is actively reabsorbed. Tadi dekat proximal, bicarbonate ion is passively reabsorbed. Kan? Okay. So NaCl actively macam biasa. So kenapa? Na plus reabsorb, then Cl followed, right, by passive transport. So overall we call as active transport lah. And also you have to remember why water is reabsorbed by osmosis passively, because of the osmolarity, changes in terms of the osmolarity in the surrounding capillary or surrounding tissue. Okay, right. Okay, so if we combine the picture, then you can see this is the cortex area, right? Ingat kidney kita ni, right? Let's say this is our nephron. All right, glomerulus. So some part of the nephron lies or confined at the cortex area, and some part of the nephron, right, lies within the renal medulla area. Okay, so I don't want you to confuse why is this one is explaining about cortex. Cortex means you can see this part, proximal and distal, confined ataupun duduk dekat kawasan renal cortex of the nephron. Okay, whereby usually loop of Henle collecting duct will be lies within the medulla region. Okay, so you can see these are the two picture combining the reabsorption process that happened at proximal and also distal tubule. All right. So to sum up, you can see this is a table that's showing you the active reabsorption and passive reabsorption that happen at the proximal and also distal tubule. So remember, at proximal tubule, we have active transportation or active reabsorption of nutrients like glucose, amino acid, right? And also salt, right? And passive reabsorption of chloride ion, okay? Bicarbonate ion, potassium ion, and also water by osmosis. And for distal tubule, we have active reabsorption of bicarbonate ion and sodium ion, and passive reabsorption of chloride ion and also water. Right? Okay. Okay. Now let's see what happened or the reabsorption process that happened at the loop of Henle. So loop of Henle is divided into two. Descending limb loop of Henle. Which one is descending? The one that you can see movement of filtrate going down. So this is descending loop of Henle. Right? And as the filtrate is travel upwards, so it is actually move along the ascending limb loop of Henle. All right. So remember, Henle must be capital letter. Okay. All right. So water and ion move differ in this region. Right. So we are going to discuss in the next video. So what happened upon the reabsorption process that happened at the loop of Henle. Okay. So see you in the next video.